Welcome to our second podcast on the digestive system. In the first podcast, we looked at the oral cavity. Now we're going to look at making our way down through the alimentary canal. And we're going to start with the esophagus and stomach. However, throughout much of the alimentary canal, we're going to follow the same generalized plan that you see on the left. So first, we're going to go through the generalized structure, then look at the differences between the regions of the esophagus and in the stomach. So we're going to find those generalized regions. We're going to identify the differences that are present in the esophagus that help us distinguish it from the other portions of the alimentary canal. And as we get into the stomach, we're going to see it's so incredibly diverse that regions in the superior portion or the fundus of the stomach are going to be different than what we see in the pylorus or the inferior portion of the stomach. So we're going to recognize those regional differences. We're going to compare and contrast gastric glands versus gastric pits. And then we're going to identify the cells that are basically present in the stomach. There are quite a few of them, and we will talk about their histology so we can identify them and their function. But first, the general plan of the alimentary canal. And essentially with this basic plan, we're going to be looking at 4 layers. You can see them in highlight here. The epithelium is going to be part of the mucosal layer, and this is going to extend from our epithelium, some connective tissue, to a thin layer of smooth muscle. Then we get into the second layer. This is the submucosa. This is connective tissue. And then we'll get into our third layer as we're moving out in our muscularis or muscularis externa, sometimes it's named. And then finally we get into the connective tissues surrounding the alimentary canal, and this is either going to be an adventitia or, in this case, they're illustrating a serosa. So starting with the mucosal layer, the one on the innermost portion of the canal, this is where we find the epithelium, and depending on where we are in the alimentary canal, it could either be simple columnar, if we're in the intestines, for instance, or it might be stratified squamous, non-keratinized epithelium in the upper part of our esophagus. And so we're going to recognize that epithelium. Immediately beneath that, so beneath the basement membrane, we're going to find the lamina propria, loose connective tissue. We have a lot of blood vessels here. Some glands might be present here in different regions. Lymphatic vessels, we might see some lymphoid nodules, not nodes now, but those primary and secondary lymphoid nodules. And then the outermost layer of the mucosa is smooth muscle. It's the muscularis mucosa. Some might call it the muscularis interna, but we're going to stick with muscularis mucosa because it's part of the mucosal layer. And while we can't readily, and we're not going to be so worried about identifying the inner circular versus outer longitudinal layer, understand those are the appropriate orientations of these 2 muscle layers that make up the muscularis mucosa. Our second layer, the submucosa, this is dense irregular connective tissue. Many of the components that we've already identified in the dense irregular connective tissue. When we look for the glands that are deeper, we saw them in lamina propria, but if they go down to the submucosa, then we're going to recognize them as either esophageal glands or duodenal glands, and we'll point those out when we get there. It's one way to help us distinguish where we are in the alimentary canal. We'll have many blood vessels, lymphatic vessels, and this is where we are going to have a special nerve plexus or network, and this is called the Meissner's submucosal plexus. It's part of the enteric nervous system and the autonomic nervous system. And so in Meissner's, we are going to be functioning for the motility of the mucosa, to a lesser degree the motility of the submucosa, but the primary reason, secretion of glands. Now, this particular plexus, part of the nervous system, this is going to control very local conditions near where these nerve plexes are present to begin with. We have a deeper one that's going to be more of a global, broader control center, and we'll talk about those as we get there. So in this particular illustration, we are looking at the mucosal layer. Here we have on this side, we have our epithelium. We come down, we've got our lamina propria, which is all sort of mixed in here. This is going to be our muscularis mucosa. You can see this sort of wavy, smooth muscle. It's very thin. Then we've got our dense irregular connective tissue, and it's within this submucosa region that we find these nerve plexes, and you can recognize these large nuclei, nerve cell bodies, and nucleolus in the middle. So these are our nerve cell bodies, and this is going to be our submucosal plexus. Now the third layer is our muscularis externa. This is extremely thick, smooth muscle. And what we're going to find is the inner portion is going to be circularly arranged, so it's going to constrict the lumen. And the outer layer is going to be longitudinal. And when it contracts, it's going to bring the regions closer together. And both of these are going to function in the process that we know of as peristalsis. Now, it's between the two muscle layers where we find our second enteric nervous system, part again of the autonomic nervous system, and this is the Auerbach's myenteric plexus. Now this is going to function on a more global scale because it's going to control a broad range of conditions, not just local but also extended further away, so you're controlling more of this longer alimentary canal, especially as we get down to the much longer intestinal tract. So in this illustration, we can see our smooth muscle. The circularly arranged smooth muscle is on top. You can see the cross sections here. And then our longitudinally arranged smooth muscle is going to be in this field. And situated between, we have these structures, and this is going to be part of our enteric nervous system. And since we're situated between these two thick bands of muscle, we know we're in the muscularis externa. And so we know that this is going to be our myenteric plexus. Now, the fourth layer is connective tissue, and this is going to be termed an adventitia if only the connective tissue surrounds it. Now, in our alimentary canal, we may actually be attached to the body wall and so lie underneath the peritoneum, in which case we are retroperitoneal, or interperitoneal, and this is where we have this outer covering, which is a mesentery. It's part of the mesothelium. And so this mesentery covers over the alimentary canal and forming our visceral peritoneum, and in which case this adventitial layer is now called a serosal layer. So once again, in our illustration, we can basically see the mesentery here covering over the connective tissue part here, and this is going to be our serosa. Now, we've mentioned the enteric nervous system. We talked about the Meissner's submucosal, and we talked about the Auerbach's myenteric plexus. Well, again, these are part of our autonomic nervous system. Parasympathetic and sympathetic control here are going to control peristalsis during the parasympathetic response, and it's going to be shut down during a sympathetic response. In this illustration where you see the yellow, these are our submucosal, the deeper portions that we see here, the Meissner's submucosal plexus, and then between our smooth muscle, here's our circular, here's our longitudinal, this is our muscularis externa, here we have the myenteric or Auerbach's myenteric plexus. So remember with the parasympathetic, this is the rest and digest. So we're going to stimulate peristalsis, we're going to inhibit the sphincter muscles so material can pass through and secretion is going to be ramped up. However, with a sympathetic response, we are going to shut down the system. The sphincters are going to close, peristalsis is going to be inhibited, and most of our blood is going to be diverted away from the digestive tract and alimentary canal, primarily to our heart, lungs, and skeletal muscle. And now to the esophagus. We're going to take that generalized plan for alimentary canal, and we're going to specialize it for whatever reason we happen to be in. And the esophagus is the first part after we leave the oral cavity. So it's going to represent the first 25 centimeters or so of the alimentary canal. Now, as we bring food into the mouth, we are going to masticate it, the process of mechanical digestion. We're going to add saliva, so the chemical digestion, and we're going to produce what is referred to as a bolus. This is just a mass of material. And so for food, this is a spherical mass, and depending on how big your mouth is and how much you like to swallow at a time, it's going to be anywhere from probably the neighborhood of 2 centimeters in diameter. And so this bolus is going to be moved from our oral pharynx down toward our esophagus through this tube that we call the esophagus. Now, the process of moving this food is going to be peristalsis, and it's going to move at about 50 millimeters per second. Now, the upper third of the esophagus is going to be controlled largely by skeletal muscle, the lower third by smooth muscle, and the middle third is going to be a mixture 50-50 of smooth and skeletal muscle. So that's another way that we can distinguish what region of the esophagus we're looking at if we happen to see a cross-section of the esophagus itself. Now, part of the muscle that we have surrounding the esophagus, part of our muscularis externa layer, is going to give rise to physiological sphincters. Now, at the beginning, we have the pharyngoesophageal sphincter, which really is the pharynx itself, part of the epiglottis and the structure of the pharyngeal compartment. But as we get lower in the esophagus, before we get to the stomach, we have the gastroesophageal sphincter, often referred as the lower esophageal sphincter. And that really is going to be the controlling structure that's going to prevent the acid in the stomach from regurgitating back into the esophagus. If that happens, we refer to that as heartburn, or we also refer to that as reflux. So as we look at the mucosa, this is going to be stratified squamous non-keratinized epithelium. And typically when we look at a cross-section of that epithelium, the cross-section is going to be collapsed, the lumen. And so when we see this folded, collapsed lumen, many branches, that's going to be indicative of what we can think of as the esophagus. Now, as we look at the cells that are present in the submucosa and the mucosal layer, we're going to find a lot of Langerhans cells. So this is similar to what we might find in the skin. And these are going to be antigen-presenting cells, very much part of that macrophage lineage of cells. Now, the lamina propria underlying the epithelium is where we're going to find esophageal glands. Now, they're called esophageal cardiac glands simply because of the location of the heart relative to the location of esophagus and the heart in the thoracic cavity. Now, these glands are going to be mucus producing to lubricate the passage of the esophagus. The food can often be very dry. And if you've ever tried to eat something that's very dry, you're going to have a hard time going down. So you'll drink some fluids, but the mucus is going to help to do that as well. Now, we do have a cardia of the stomach, the cardiac region of the stomach, and in some ways these glands are going to resemble those as well, although not nearly as extensive as what we'll see. Now, these esophageal glands are going to be near the pharynx, so that pharyngoesophageal sphincter, as well as down toward our lower esophageal sphincter, and that's where we'll find these locations for our esophageal glands. Now, the muscularis mucosa, we do not have an intercircular outer longitudinal. We only will see the single layer of longitudinal smooth muscle as part of the esophagus itself. So here we're looking at a cross-section of our esophagus. We can see the folded nature of our mucosa. It's not just one big circle here. We've got our epithelium, stratified squamous. We've got our lamina propria. We really have a hard time seeing the muscularis mucosa. It's almost like a discontinuous or dashed line layer, but I think you can see these darker cells in this region. That's a smooth muscle. Then the bluish tissue we see, that's our submucosa, and then we get into our muscularis externa. And in this particular section, they're showing an intercircular and an outer longitudinal. You will not see both of these in every section through esophagus. And then with the esophagus, we are going to have an adventitia to the outside. Our submucosa, dense, irregular connective tissue, we will in fact have esophageal glands, and these are the tubular glands that we find, and they're going to have mucus cells and serous cells that are present here. Again, the mucus cells, again, secreting for lubrication, but the serous cells are going to be producing lysozyme, pepsinogen. So you've got essentially an antibacterial enzyme, the lysozyme, and you've got a protease, the pepsinogen, that's going to, again, continue the process of disinfecting, continue the process of the chemical digestion of the material that we've swallowed. The muscularis externa, as I've mentioned before, you're going to be able to see the skeletal muscle versus a smooth muscle. Upper third, 100% skeletal. Lower third, 100% smooth muscle with a 50-50 mix present in the middle. So here we have another section of our esophagus. Here we're going to have our mucosa, epithelium, lamina propria, muscularis mucosa, again discontinuous, submucosal layer, then we have an intercircular, outer longitudinal. And if we could get a better look at these, we would be more able to determine if these were smooth or skeletal muscle layers that we're looking at. Here's a little bit closer magnification looking again at our muscularis mucosa of smooth muscle, then our submucosa of our dense irregular connective tissue. The outer layer is going to be covered by the adventitia. However, as we pass through the diaphragm, remember our stomach is below the diaphragm, so we're going to pass through an opening in the diaphragm, which is called a hiatus. And once we pass through the hiatus of the diaphragm, then the esophagus is going to be covered by a peritoneum, so we will refer to it at that point as a serosa. Now, we've made it from the oral cavity down the esophagus, and we're arriving at the stomach. And this is going to be the most distended or capable of distending portion of the alimentary canal, and it can actually accommodate 1.5 liters of material. And again, this is sort of a generalized term that we give here. Some people can have their stomach expand larger than that, and some may not be able to get half of this down, depending on the size of the individual. But now, after the bolus of material has moved through the esophagus and into the stomach, we are going to add material to it, not the least of which is hydrochloric acid and other materials. And now we were going to refer to this as chyme. So it's no longer a bolus, but chyme is the product of chemical and mechanical digestion. So in our stomach, we're going to have 4 regions. The cardiac region, or the cardia of the stomach, is a very small portion where we have the gastroesophageal junction. Then the stomach is going to have this dome-shaped, almost think of it like a humpback on the stomach, and that's going to be referred to as the fundus of the stomach. Then the bulk of the stomach is the body, or the corpus of the stomach, and then we're going to get to the inferior end of the stomach, and that's going to be a narrowed funnel type region that we refer to as the pylorus. Now, the pylorus is the region of the stomach. Some people may use that term pylorus in place of the pyloric sphincter. I don't like to do that. The pylorus, I refer to this region of the stomach because the pylorus is actually the opening that's going to narrow as you approach the duodenum, the first part of the small intestine, and it's within the pylorus where we have this outer thicker muscularis externa. The middle layer is going to be much, much thicker, and that's going to comprise the pyloric sphincter. So the pyloric sphincter is a very distinctive feature present in the pylorus of the stomach. So here we can see in our illustration of the stomach, we've got the cardia of the stomach where the esophagus is coming in, the fundus, the humpback part of the stomach, the body or the corpus, and then here we have the pylorus of the stomach, the pyloric canal, they refer to it. This is the antrum of the stomach, and there you can see the thickened muscularis externa pinching in, creating the pyloric sphincter. Here we have an illustration of the opposite end. Here we can, and I think even at this magnification, we can see the stratified squamous epithelium look. We've got our papillae projecting up in there, like our dermal papillae. And then when we get to this region, this is going to take over, and this is going to be the cardiac glands of the stomach. And so this is going to be our esophageal gastric junction. Now, generally, as we look at the stomach, a little bit of gross anatomy, if you will. When you open up the stomach, you're going to see a lot of folds. Now, these are going to be indicative of a stomach that is empty. And so these folds are going to be gastric rugae. And basically, when you try to, fold up a large balloon, you're going to have these creases and folds that you produce when you get it into a small space. So these gastric rugae are going to be folds of the mucosa and submucosa that will allow the stomach to really stretch and distend when it comes full with that 1.5 liters of material. Now, the epithelium of the stomach is not necessarily continuous. It is, but it's not a flattened surface. You're not going to be inside the stomach and look at the wall of the stomach and see a solid floor. It's going to look like it's completely pitted with holes throughout. And these holes, or foveolae, are openings of what we call the gastric pits. Now, the epithelium is going to be folded downward in toward the lamina propria, down toward the muscularis mucosa, and these openings, these pits, they're going to lead to the bottom of these invaginations. You're going to have different cells in the very beginning of the pits, and the cells are going to change and become different cells as you get to the bottom, and there we refer to them as gastric glands. So imagine poking your finger down in the epithelium and making this deep invagination, so that up by your knuckle at the beginning of this created invagination, you're going to have a certain set of cells, and down at your fingertip, the cells are going to be different. And so these are gastric glands. Now, these glands are going to branch off. They're tubular glands, but they're going to branch off from one pit. And so they will empty the materials into that one pit. So here's an illustration showing our stomach. We're not really seeing the gastric rugae. We could see that in the illustration previously. But if we look at the wall of the stomach, here are these pits, these invaginations that you have. And they're going to extend down. Here we can see the epithelium. The epithelium will invaginate in. But as you get deeper and deeper, you can see that these colors change. These are going to represent different cells. And if we can look at one of these pits versus glands, here's what we're going to see. So here's the opening or the pit. We're going to have the isthmus, the neck, and the base. But the gastric gland is going to be the secretory cells toward the bottom. So we're going to have surface lining cells, and that's what we refer to as our epithelium in the stomach. But then we're also going to have cells that produce mucus neck cells. We're going to see some fairly large rounded cells that are present in this region, sort of in the middle or the neck region. And these rounded cells are going to be oxyntic cells. Oxyntic means we're producing acid, hydrochloric acid, and these are going to be the parietal cells. Now, as we get toward the base, we're going to find cells that are zymogenic cells, and a zymogen is an enzyme. So these are more serous producing cells in general, and they're secreting enzymes, and they're referred to as chief cells. Oxyntic and zymogenic really are functional terms. Parietal and chief are more proper names of these cells. Now, in the very base, we're going to have a number of these DNES cells. They're enteroendocrine cells that are producing quite a bit of hormonal substances, and we'll see those down in the base. So here we have a cross section through our gastric gland. Here we can see our surface lining cells, and you can see how those are invaginated downward. This is going to be the beginning or the neck of our gastric pit. Here you can see a pit coming down, and you can actually see the branching nature of this one. Now, as we get deeper, you can see how these cells, some of them are very rounded in nature. Those are going to be our parietal cells, and here you can see this fat parietal cell that we have here. Now, as we get deeper and deeper, when you see these cells that have all these little vesicles present in them, especially down at the base, these are going to be our zymogenic or chief cells at the base of our gastric gland. Here you can see the muscularis mucosa, and so these gastric glands extend very far downward toward the muscularis mucosa, with our lamina propria extending all around these gastric glands. So now, as we talk about the different regions, as always, we're starting with the mucosa. And so now we are almost exclusively, we've done away with the stratified squamous. Now we're simple columnar, and we'll maintain this throughout much of the rest of our alimentary canal until we get to the end, and we'll then change back to our stratified squamous type epithelium. But these are our surface lining cells, and you'll see that SLC abbreviated throughout a lot of the literature. And these surface lining cells are actually secretory, and they're going to produce what is referred to as visible mucus. Now, this visible mucus is going to adhere to the surface of the cell, and it's going to protect the epithelium from autodigestion. And the way it does that is this visible mucus produces, these cells produce bicarbonate ions, and they trap those in that mucus layer. So when the hydrochloric acid encounters the bicarb, it neutralizes the acid, and therefore it does not destroy the cells. Now, as you can imagine, as these parietal cells, oxyntic cells, are producing hydrochloric acid, emptying it into these glands, into the pits that empty into the stomach, you must have a very tight seal so that this material does not get out of the stomach. So the surface lining cells, and really all of the cells that are present in our glands and pits and on the lining surface, they have zonule occludens and zonule adherens, those structures that we're comfortable with already with the lateral boundaries of our epithelial types of cells. Now, we also have regenerative cells. I think that was listed in our illustration, but these regenerative cells are important because as these cells are exposed to this harsh environment, you've got the very likelihood of having a mutation. And so these cells are going to be replaced about every 5 to 7 days. So once a week, you're going to turn these lining cells over and produce new cells to prevent them from accumulating too many mutations because of the acidic environment, the environment of all the friction and material that's present there. So the regenerative cells, they're going to look a lot like their surface lining cells. They're columnar in nature. You have the zonule occludens, zonule adherens, but these are going to be highly mitotic. And so if you happen to see a figure and you see mitotic activity, you know that's a regenerative cell that's becoming a surface lining cell. Now, the glands and the pits, as I mentioned, extend downward into the lamina propria, and any stomach is going to have about 15 million gastric glands present. So you have a lot of these glands and a lot of these cells producing a lot of hydrochloric acid and enzymes. So we're going to look at the fundic, because in the stomach, these glands don't look exactly the same in different regions of the stomach, the cardia, the fundus, the body, and the pylorus. So we're going to look at these fundic glands. They're going to be fairly similar to what we'll see in the body, but they're different than what we're going to see in the pylorus. So our fundic glands, we divide those into the isthmus, which is the first part of the gland right at the neck, and it's mostly our surface lining cells. Then we get to the neck, the middle zone, and this is where we have some of the surface lining cells, but more likely the mucous neck cells, the regenerative cells, parietal cells. We may have some DNES, but most likely those DNES are going to be at the bottom. So here we have another illustration showing our pits. These are much more open as we see them extending down. And again, most of what we see here are going to be our surface lining cells. As we get deeper, we're getting into our parietal cells. We've got our regenerative cells. Our chiefs and DNES are likely going to be down here at the base of our gastric glands. And here we have our muscularis mucosa that ends our mucosal layer of the stomach. This is a neat stain that we have here of these cells. Here we have our lining cells that are producing mucus, and so this is some of the mucus material that's present in these lining cells. And you can see these much more rounded, larger cells, and these are the parietal cells as we get down into the neck and the middle portion of our gastric pit and gland. Now, again, at the base, this is where we're going to have mostly chief cells. We'll have DNES cells, may see some parietal cells down here, but principally we're going to be looking at our chief cells. So down in the base, we can see, again, these rounded parietal type cells. And again, these are oxyntic in nature, producing the hydrochloric acid. These darker cells are going to be our chief cells, our zymogenic cells. And then sometimes we have these lighter cells down here, smaller in nature, certainly not big like we see with the parietals. And these are going to be our enteroendocrine, or the DNES cells. So now let's look at and talk about the different cells that we have and how they differ in their function. The mucous neck cells, of course, they're producing the mucus, and this mucus is going to be for lubrication. This is more like our goblet type cells produced here. These are not the bicarb visible mucus that's produced by the SLCs. Now, these are columnar cells, apical microvilli producing these, and again, similar, the product is like from goblet cells, but these are not going to look like goblet cells. They're just mucous neck cells, and we are going to have our tight junctional complexes, so the material is not going to be able to spill past these cells and the other types. These are all of our epithelial types of cells present here. The other cell that we're going to see, the large parietal or oxyntic cells, these are going to be the easiest ones, I think, to recognize just because of how big and rounded they are. And these are going to produce hydrochloric acid, and they're also producing gastric intrinsic factor. Now, this one is important when we talk about pernicious anemia. And so this B12 is going to be important for the production of red blood cells, and intrinsic factor is essential for the efficient absorption of B12 out of our diet. And so this intrinsic factor is produced also by our parietal cells. Now, the way HCl is produced, and we're going to get to this toward the last part of the chapter, but it's not produced intracellularly. Portions are produced and secreted out. You have chloride ions secreted, you have hydrogen ions secreted, and they come together externally to produce hydrochloric acid. But you're going to have these deep invaginations down into the cell. And these are referred to as intracellular canaliculi. These are just enfoldings of the plasma membrane down into the cell. So you create this environment sort of within the cell that is really external environment where this material can simply flow from the canaliculi into the gastric gland. It's not actually inside the cell. And so when we look at a picture, it'll make it a little bit easier to recognize that. Now, these canaliculi have microvilli, in this case not for absorption, which we usually think of for microvilli, but to increase the surface area for secretion. So, as microvilli number increases, that's going to correlate with increased hydrochloric acid activity. Also, the vesicle number of the substrates that we're using, that's also going to increase as hydrochloric acid activity increases. So here we have a parietal cell, and even in this one, we can recognize this nice, rounded, large shape that we have. Here is our nucleus of our parietal cell. Tons and tons of mitochondria, because we're going to be pumping this material out of the cell. And here we can see one of those invaginations in the microvilli of one of these intracellular canaliculi. It goes out of the plane of section here because we have cytoplasm, but then we pick up the other part down here. This is another part of that canaliculi in the microvilli. This is happening all around the nucleus three-dimensionally. So it's almost like you have this island that's the nucleus. You have this outer casing of cytoplasm, and then you've got this canaliculi that goes all the way around. Here you can see other sections through the canaliculi running through the cell. I believe we have an illustration toward the end where we talk about hydrochloric acid production, and it'll make it a little bit easier to grasp that concept. Now, the chief cells are the zymogenic cells. I think some have called them peptic cells. These are producing the enzymes pepsinogen, renin, gastric lipase. All of these are going to function in the process of chemical digestion and the breakdown of food. So when you get to the base of the cell, if they're not the DNES cells, they are going to be chief cells. They're going to be darker staining, a lot of granules, and these granules are filled with enzymes. Now, since these are enzymes, it's not surprising when we look at these chief cells, there's a ton of rough endoplasmic reticulum. They're producing proteins, secretory proteins, and here we can see these zymogenic granules, these vesicles that are beginning to get close to the surface, and will secrete them into the base, into that lumen at the base of the gastric gland, and that will flow into the lumen of the stomach. Now, renin in this case, it is an enzyme. It is a proteolytic enzyme, and it's called chymosin. And in this case, this particular enzyme's chief function is to degrade milk protein. Now, milk would flow quickly through your stomach if it were in a liquid form. And so renin is going to curdle the milk. It's going to denature the milk proteins. And so it's going to convert this liquid into a semi-solid, almost like cottage cheese type material. And that's going to be retained in the stomach for longer periods of time. Now, we don't secrete this enzyme. I mean, we don't produce it and it's not active intracellularly. It's secreted as prochymosin because it's not going to be activated until it hits the acidic environment in the stomach, because most of these enzymes that are present there would be degraded and denatured by the enzymatic activity, by the low pH, but they're designed to be active there. So we keep them inactive as we produce them, secrete them. That low pH activates them by removing parts of the protein, and therefore they can do their work. Now, these last DNES cells, diffuse neuroendocrine cells, also called enteroendocrine cells, because of their products that they produce are hormone-like in nature, and there are 13 different types, and we're not going to get into all of the different types. We're going to label a couple of them. But basically what we're going to see in category-wise, these cells can either be open or closed. Open in that they extend from the basal lamina all the way to the lumen, or they can be closed. In this case, they're going to be like the regenerative cells where at the base, but their superficial surface doesn't reach the lumen. And so in that case, they refer to as closed. Now, the way they produce and secrete their materials, it can be paracrine, a local type reaction, endocrine in nature, because they're close to the lamina propria where there are a lot of blood vessels. Those secretions are picked up by the bloodstream. Or sometimes, here's a new one, since their secretions are similar to neurotransmitters, sometimes they're referred to as neurocrine. That's one you won't hear quite as often. But the different DNES cells we have are A cells. They produce glucagon. We're familiar with that. ECL cells produce histamine. The G cells produce gastrin, and this particular hormone is going to stimulate increases in hydrochloric acid production. And then we have I cells. I cells produce cholecystokinin. And cholecystokinin is going to stimulate the release of pancreatic hormones, but cholecystokinin is also going to stimulate peristalsis of the gallbladder. If you have to go in and have a gallbladder function test, the way they're going to cause your gallbladder to become active is they're going to provide you with cholecystokinin and then see how your gallbladder behaves. So once again, we're looking at gastric glands and pits. Here we have the pits on the left extending downward. I think you can already pick out the large parietal cells that we have. And as we get toward the base, we're going to be expecting to find more of our chief type cells that we have here. Couple of parietal cells, and then here we have one of our DNES cells just sort of present in here. Doesn't really look like much of any other cells that we have. And again, most of what we see are going to be our chief cells. All right, our muscularis mucosa, as we get toward the end here of our mucosal layer, we are going to have an expanded mucosa. We've got our inner circular, middle longitudinal, and may have an outer circular, although this is not going to be the thick layer. Our thick layer is most definitely going to be the outer muscularis externa, but we do have a more expanded muscularis mucosa present here. Now, before we get any deeper, we're going to look at the differences. And so the difference is largely between cardiac and fundic and pylorus. Here's some generalities. When we look in our gastric pits, they are going to be shallower and the base of the glands are going to be coiled in the cardiac region. So in the pyloric region, they're going to be much deeper. They may go halfway down to the muscularis mucosa. Now, in cardiac glands, we're not going to have any chief cells. We're pumping out the hydrochloric acid there very much. And when we get to the pyloric glands, what we're going to see is the predominant cell type is going to be the mucous neck cell. Because we're producing mucus, we have to protect the lining cells here, and we're trying to neutralize as much of the acid as we can before we go into the intestine. So you're going to see many more of those mucous neck cells in the pyloric region. In the pyloric region, the glands are going to be much, much deeper than they will be in the cardiac region. So in this case, we can see our gastric glands and pits. There we can see our muscularis mucosa, submucosa, and our fairly thin muscularis externa. Now as we get here, even thicker muscularis externa. Here we can see our gastric pits and gastric glands. Where can we see our muscularis mucosa? And there we see our submucosa. Now, if we could look even closer to look and see where the pits and the glands are, we would be able to determine if we're in the cardiac or the pyloric regions, or if we're somewhere in between. Now, the submucosa is going to look pretty much what we've seen before. We're going to see our dense irregular connective tissue. The muscularis externa, we have 3 layers in the stomach, as we saw in our diagram, our illustration. We have an inner obliquely arranged muscle. Now that's going to be new compared to what we see everywhere else. And we have a middle circular and an outer longitudinal. So we're adding that inner oblique to our generalized plan. Now the middle circular, this is the one that's going to be expanded and form our pyloric sphincter in the pyloric region. And then in the outer region, we're going to have a serosa, and that covers the entirety of the stomach. So here we can see that we've got our gastric glands, we've got our mucosa, we've got our submucosa. Submucosa is here, our muscularis is going to be here. This is our muscularis externa, and this is the pyloric sphincter. Much expanded middle portion that we have here projecting way up, causing this huge ridge to be formed. And then after we leave our gastric pits and gastric glands, our mucosa is going to change drastically as we get into our intestine. We have a ton of glands that are going to be present here in the submucosa, and this is going to be indicative of moving into the duodenal portion of our small intestine. Now, hydrochloric acid production, we sort of touched on it as we talked about our parietal cells. But what we're going to do is our parietal cells, or our oxyntic cells, are going to be responsive to gastrin, histamine, and acetylcholine. Remember, acetylcholine is secreted by those post-ganglionic parasympathetic neurons, and so that's going to activate this as we ramp up our digestive tract. Well, intracellularly, like we talked about with bone, we're going to have this enzyme, carbonic anhydrase. It's almost as widespread as the sodium-potassium pump, it seems. But carbonic anhydrase is going to first produce carbonic acid, and it's going to do that from carbon dioxide and water. We're going to break that down into our hydrogen ions and our bicarb ions. Well, the bicarb ion, that's going to be picked up by our surface lining cells and used as part of that visible mucus. But these parietal cells have a hydrogen ion, potassium ion antiport. So out goes hydrogen, in goes potassium, right? We're going to keep potassium high on the inside. Now, what we're going to do to keep those potassium levels from getting too high, we're going to have separate pumps. And these other pumps are going to pump out the potassium ions. Now, we also have a third type of pump, and these pumps are going to pump out the chloride ion, and it's going to pump these out into those intracellular canaliculi. So as the chloride ions get pumped out, the hydrogen ions get pumped out, they will combine together to form hydrochloric acid. So here we have one of our parietal cells at rest, and here you can see they refer to this as tubulovesicles, tubulovesicular apparatus, and we've cut this in cross section, but understand this is going around. So we have somewhat of a cone shape in the middle surrounded by the walls of our cells. So it's almost like a moat surrounding that. Now they've removed sort of this middle portion to give you an illustration of what's happening, but here's our chloride pump. Here we have a potassium pump. And here we have our hydrogen-potassium antiport. So this pump is only keeping those potassium ions recirculating so that we can pump out the hydrogen. As you can see here, that's going to require energy. But as we do this, the hydrogen and chloride ions produce the hydrochloric acid outside of the cell. And when the cell's actively pumping, here you can see those canaliculi that are created, those passages where the hydrochloric acid is actually outside the cell, even though it's deep, and that can diffuse out into the lumen of the stomach.